I apologize, everyone. I got ahead of myself and forgot to show you the images for the mechanisms of action. So let's go over those. So at the top here is what the cell wall is going to kind of look like. So the little circles coming down are NAG and NAM. Okay, so there's our glycan strands. And then if you look at picture 2, you'll notice that the little yellow lines that were originally pointing outwards in opposite directions are now being catalyzed to join together. So now the right two glycan chains are connected. And that's what we want. That's going to make a nice, strong cell wall. And so this enzyme here is responsible for making that happen. Well, if you notice, penicillin is going to come in here, and it is going to be able to bind to this enzyme, okay? And it is going to prevent further cross-linking from happening, okay? Because ideally, the bacteria want all of these glycan chains to be linked together, okay? But once penicillin is entered into the equation, it is able to competitively inhibit, or it's said weird, it is able to bind to this enzyme, which is a form of competitive inhibition. And so the enzyme can't come in here and do its job, okay? Here is bacitracin. And so remember, this is the one that I said says get back. It doesn't allow the peptidoglycan subunits out of the cell. And so here's NAM, which needs to get outside of the cell. So down here on the bottom is outside of the cell. Oops, sorry. And the cell is trying to transport NAM outside of the cell so it can build that cell wall. But you'll notice bacitracin is here at that transport protein, and it's blocking it. It's not going to allow NAM to exit the cell, which means that cell wall cannot be completed. Okay. Now here with vancomycin, notice on the left, the optimum goal here is this upper layer of NAG and NAM is going to be linked to the bottom layer by these 5 pink circles. That's our peptide links. And so what vancomycin does here, as you see in the middle, is it actually binds to the glycan chains and it prevents that peptide linkage from being formed. Okay, so again, it's disrupting the integrity of the cell wall, so it's going to be osmotically fragile and can't withstand that trigger pressure. All right, now we're really done with the cell wall synthesis inhibitors.